It appears like this. And here all these small structures to the dwarf shoot, and from here needle-like leaves are developed. These are the leaves. These are the leaves developed here. You can call it the long shoot. These are the bluish one is the dwarf shoot. And these are needle-shaped leaves. It's a pine tree. And at the place of origin of dwarf shoot is short-lived. Dwarf shoot is always short-lived. And at the place of origin of dwarf shoot, male cone, and at the place of origin of secondary shoot, you see, this is the secondary long shoot. And here you will get the female cone. Now, first of all, you see one thing, that the leaves are needle-shaped. And what is the advantage of these needle-shaped leaves? Leaves are needle-shaped. Suppose, even I am going to show you in detail, it appears like this. Suppose this is the dwarf fruit. Suppose this is the secondary long shoot. And here you will get the leaves. So this is the secondary long shoot. This one is dwarf shoot. And these are the leaves. So I think it is clear now. And why leaves become needle-shaped? Basically, you know, pine trees are found to be present at least 3,000 feet above the sea level. That's why in Tripura, pine trees are not available. You'll get it in Shillong. If you go through the routes of whenever you have to actually travel from Guwahati to Shillong, whenever the height is almost 3,000 or more, gradually you will be seeing this pine tree. Because this is the height. For this, they have to face some extreme conditions, extreme temperature, extreme actually humidity and wind. And to overcome this problem, they have some adaptive features. What are these adaptive features? First of all, as they have to face all these conditions, what is required? The rate of transpiration is required to be decreased. They have to check the rate of transpiration. And for checking the rate of transpiration, the surface area of the leaf is required to be decreased, and hence the leaves are needle-shaped. You know, in a needle-shaped leaf obviously surface area is much more lesser, so that the rate of transpiration will be less. Second one, the leaves are found to be covered with cuticle. Cuticle also protect the leaves, and also it prevents the rate of transpiration to some extent. Thirdly, in their leaf, stomata are found deep-seated, much more inside the leaf, and such stomata are called sunken stomata. Sunken stomata are found. Sunken stomata are found, and that's why they can check the rate of transpiration. What I said, as their leaves are needle shaped, so the surface of leaf decreases, so it can check the rate of transpiration. Secondly, the leaves are protected by cuticle, so it also to check the rate of transpiration. And thirdly, the stomata are not exposed like the other angiospermic plants, rather they are found to be much more inside the leaf, and such stomata are called deep-seated stomata, and these deep-seated stomata are called the sunken stomata. And sunken stomata obviously will reduce the rate of transpiration. Is it clear up to this? Is it clear the adaptive features of pine tree?
Yes, sir.
Okay. Then comes to the its cones. You know, in the pine is monoecious. In the same plant, you will get the male cone and female cone. Like this, you see. These are the female cone. And they are the male cone. Found present in the same plant. Later on, if you separate them out, this is the female cone, the male cone, and female cone. Then Sumanta Saha. In cross and longitudinal section, it is the male cone, and at the base, next, in case of female cone, in LS, within cross section and here at the base, you will get an anatropous ovule. So these are called microsporangium, and it is called ovule. Inside the microsporangium, you get microspore mother cell. Inside the ovule or megasporangium, you will get megaspore mother cell. After that, microspore mother cell undergoes meiosis. To form initially spore tetrad, and later on the spores are separated out. Outer coat called axile and inner one called intile, and this is the microspore, and this is spore tetrad, and megaspore mother cell also undergoes meiosis. To form functional megaspore. Then it undergoes mitosis to produce more than one archegonia and the endosperm. And the endosperm is haploid. Inside the archegonia, you will get one egg and one small ventral canal cell. So you can write archegonium. This is the egg, and this one is ventral canal cell. Eventually, this is the ovule. Here you will get the pollen chamber, and in the meantime, the outer wall of the pollen grain you can flatten to form winged pollen grain. And it is 4 cell stage. The cells are first prothelial cell, second prothelial cell, the vegetative cell or tube cell, generative cell. Now it is transferred to the ovule, and the process is called pollination. After reaching there, the generative cell divides to form 2 male gametes. Here you will get the egg, and before fertilization, ventral canal cell degenerate. So eventually after that, 1 male gamete and egg fuse to form zygote. Zygote to form embryo, to form so the ovule, after fertilization, ovule gets converted to, after fertilization, ovule gets converted to seed. That means you can idea. Converted to seed. So zygote is formed, embryo is formed inside the seed, and eventually, secondary shoot, young prophytic plant which is also deployed in occurs. So this is one of the life cycle pattern found in Pinus. And as more than one embryo formed in a single seed, the condition is called polyembryony. Another most important thing about Pinus is polyembryony is noticed. Because in one seed, more than one archegonia means more than one egg, means more than one zygote, means more than one embryo. In this way, in one seed, more than one embryo is formed, and the condition is called polyembryony. And although a number of embryos are formed, ultimately one embryo is found to be viable and will be giving rise to new sporophytic plant. Deva বুঝতে পেরেছো কিনা? আমরা
স্যার খালি একটা এমব্রয়ডারি হবে না যে এই নতুন প্ল্যান্টকে জার্মিনেট করবে যে
একটা মিনিট। হ্যালো,
স্যার এই অনেকগুলো এমব্রয়ডারি থেকে তো খালি একটা এমব্রয়ডারি ওই মানে নতুন প্ল্যান্টকে জার্মিনেট করবে না।
একদম রাইট।
হ্যাঁ এটাই স্যার।
আচ্ছা, এখানে আমি একটু অ্যাড করে দিচ্ছি। অ্যামব্রো থেকে আমরা এখানে দিচ্ছি, অ্যামব্রো থেকে সিড দিয়ে দিলে সবচেয়ে ভালো হবে। ম্যাচুরেশন অফ সিড, এরপর দাও সিডলিং, ইয়াং সিডলিং 2N, seed also place it 2N, and secondary long should be there. Ultimately, the mature sporophytic plant 2N. দেখো, এটা দেখো, কোন ডাউট আছে কিনা তুমি আমাকে বলো। কিং অফ সিডা রয়েছে না, এগুলো তুমি জাস্ট একটু একটু করে পয়েন্ট না হলে আমি দিয়ে দেবো তোমাদের, ইম্পর্টেন্ট নয় আর কি আমাদের। আচ্ছা, এখানে একটা টার্ম আছে, এটা লিখে দাও তো, হোয়াট ইজ মিন বাই সালফার শাওয়ার। হোয়াট ইজ মিন বাই সালফার শাওয়ার, আমি বলি একটু শোনো। Actually, pollination time in case of pinus is almost March, April, and during the period of time from the male cone or this large number of pollen grains are liberated out as they are anemophilous, so they are to float in the air to reach the female cone. Because the pollen grains liberated from the male cone are not going to be deposited in the female cone produced in the same year, because female cone requires 2 years to develop. That's why the pollen grains liberated from the male cone produced this year are to be deposited in the female cone produced last year. That's why they are to float in the air, and then a lot of pollen grains which are yellow in color are to float, and most of them are falling down, and it appears like that of the shower, and hence the term used is sulfur shower. Why sulfur? Because sulfur is yellow in color, and these pollen grains are also yellow in color. Is it clear to you? Basically, in March, April, the pollination time in case of pinus, and in pine forest, what it is found, a large number of pollen grains are liberated from the male cone to reach the female cone produced in the earlier year or last year. And during this period, as they are anemophilous, so they are to float in the air. And while floating, what happens? A number of pollen grains are falling down. So it appears like that of shower of yellow colored pollen grains, and hence the term used is sulfur shower. বুঝতে পেরেছিস? Pinus. Now, 2-3 things you need to remember here. Point 1, gametes are not flagellated here, male gametes. Secondly, pollination takes place at 4 cell stage, most important point. Thirdly, endosperm is formed before fertilization, hence it is applied. Fourthly, out of 2 male gamete, 1 male gamete is utilized, other degenerate. Okay? And polyembryon is noticed, as more than 1 archegonia are found to be present in each ovule. মনে থাকবে? Point 1, you did have to think to remember. Point 1, pine leaves show xerophytic adaptive features. Point 1, I should be. Pine leaves show xerophytic adaptive features. ফিচারটা বললাম না, গীতা আমি আগেই বলেছি। Point 2, pine is a monoecious plant. That is, male cones and female cones are produced in the same plant. Pollination, Pinus is anemophilous. Pinus is anemophilous means when pollination takes place with the help of air. And pollination takes place at fourth cell stage of pollen grain. As endosperm, next point, as endosperm is formed before fertilization, it is haploid. Out of two male gametes carried to the present in the pollen tube, one male gamete fertilizes the egg, other degenerate.
স্যার
Huh?
স্যার এন্ডোস্পার্মেটটা কি বলেছিলে?
Endosperm is formed before fertilization and hence it is haploid. Lastly, polyembryony is noticed. Which is formed in one seed. Pollination is actually pine is anemophilous plant. Anemophilous means the pollination takes place by means of wind. And pollination occurs at fourth cell stage of pollen grain. Cells are first prothelial cell, second prothelial cell, vegetative cell and generative cell. Inside the next one, the endosperm is formed before fertilization and hence it is haploid. Thirdly, as more than one archegonia are formed in each ovule, so more than one egg will be there, so that eventually more than one embryo will be formed. As more than one embryo will be formed, so eventually you will see in one seed, when ovule is converted to seed, then you will get more than one embryo in seed, and this is called polyembryony. So things to remember in Pinus. Pinus is a monoecious plant having male cone and female cone in the same plant. Point 2, the pine leaves show some basic xerophytic characteristics like presence of needle-shaped leaves, sunken stomata, and the cuticle covering to overcome extreme conditions, extreme heat, humidity, and wind. In the male cone, microsporangium is there, where microspore mother cell produced undergoes meiosis to form the microspores. Microspores, the outer wall will form wings-like structure, so that it is called wing pollen grain. At fourth cell stage of pollen grain, pollination occurs. The functional megaspore produced in the female cone undergoes mitosis to produce endosperm and archegonia. In this way, endosperm is formed before fertilization and hence it is haploid. And then what happens next, that as more than one archegonia are formed in each ovule, so more than one egg should be there, and eventually more than one zygote and embryo will be formed. So when one ovule is converted to one seed, inside one seed you will get more than one embryo, and this condition is called polyembryony. Although a number of embryos are formed, ultimately one embryo becomes viable and it will give rise to new plant. Now, do you have any doubt? Angiosperms. Basic features, same. Main plant body is diploid and it is dominant. And leaf. Primary roots from taproot system. Adventitious roots are also found. Fourth, stem is erect. Sometimes woody, sometimes soft. It varies actually, the stem characteristics. Sometimes it is twining or trailing. Plumule part, germinating seed. And this root is developed from the radical part. Look one thing. If it is germinating seed, this is the radical, give rise to root. This is the plumule, give rise to shoot. Radical give rise to root and plumule give rise to shoot. Stem is erect, twining, trailing. Stem is developed from the plumule. So this is what happened. Next, leaves are developed from nodes. That means which is shoot, so this is called node. The knot-like structure is found in the stem. Next,
Sir, stem is erect, then what you have written?
twining and trailing. Twining
Okay, sir,
and
thank
trailing. Most important characteristics. For the first time, flower is found. Next, secondary growth occurs. Vascular tissues are found. Now here two most important things you need to remember. Vascular tissue includes xylem, right? Xylem components are varying. Among the xylem component in angiosperm, xylem vessel is only found in angiosperm. Xylem components are trachea, xylem vessel, xylem parenchyma, xylem fiber. Among them, xylem vessels, remember the most important point. Xylem vessels found only in angiosperms. Remember this thing. Regarding phloem also. In phloem, sieve tube and companion cells are only found in.
Sir, it is not visible.
Okay, I'll make it visible. No, don't worry.
স্যার
হ্যাঁ।
স্যার তাহলে স্যার বাকি জিমনোস্পার্ম অ্যালগি তো না
First of all, I am telling you, in algae, vascular tissues are not found, right? In bryophytes, vascular tissues are not developed. Clear? You will get vascular tissue from pteridophyte onwards. Clear? Now, the components of xylems, trachea, xylem basal, xylem parenchyma, xylem fiber. In case of gymnosperms and the pteridophytes, the xylem component which is mainly concerned with conduction of water and minerals is the trachea.
আচ্ছা
Clear?
ওকে
And regarding phloem, for the translocation of solutes, in those plants, sieve cells are present instead of sieve tube and companion cells.
ওকে
বুঝতে পেরেছিস?
হ্যাঁ স্যার বুঝেছি এটাই বলছিলাম স্যার ওয়াটার ট্রান্সপোর্টটা বাকিগুলি থেকে এরকম
হবে কি রাইট
হ্যাঁ এটাই
Now next, another point. Obviously, secondary growth occurs in dicot plants only. You know, angiosperms are of two types. One is dicot, other is the monocot. So secondary growth doesn't occur in monocot, only in dicots. Next one is, so secondary growth occurs in dicot plants only. Next point comes, double fertilization occurs. Double fertilization occurs. This is the most important feature. Endosperm is triploid. This is another most important feature.
স্যার ফার্টিলাইজেশনটাকে দুইবার হয়।
হ্যাঁ, দুইবার হয় দেখাবো আমি কিভাবে হয়।
ওকে স্যার।
Let me in. Double fertilization occurs and endosperm is triploid. দেখো বুঝতে পেরেছো কিনা? Archegonia are not found. Archegonia are not found. Twelve. Most important thing. This is for the first time. Fruit is developed. So this is the first plant group where you will get the fruit. This is about the basic characteristics of the angiosperm. Then already I gave some stars here to remember these things. Clear?
স্যার আর্কেগোনিয়াম ফর্ম না হলে এক কোথায় স্টোর হয়?
This is most important thing. ধরো একটু দাঁড়ালে একটা মিনিট ক্লিয়ার তো না বলেছিলে আমাকে না
হ্যাঁ স্যার করেছিলাম।
হ্যাঁ বলো
স্যার ওই আর কে গুণে না থাকলে এক কোথায়
ও
স্টোর হয়?
আর কি গুণিয়ে না থাকলে এক্ষুনি আমি দেখাচ্ছি এটাকে লাইফ সাইকেলটা দেখি এর মধ্যে দেখিয়ে দিই একসঙ্গে বিকজ হিয়ার actually pistil is developed having style stigma and ovary and inside the pistil you will get the ovule and the ovule you will get the megaspermata cell. I'll be showing you then you will see why it is not liquid.
আচ্ছা আচ্ছা।
Look here. So flower. So I am coming to the flower directly. So this is a flower. The sepal. So this is what is called stamen. And it is called pistil. Now you see next. Ovary, style and stigma. Then this is called the anther and this one is filament. So in the anther you will get here the microsporangium. Pistil, ovary, inside the ovary ovule is found. Microsporangium was this microspore mother cell undergoes meiosis to form initially spore tetrad, later on spores are liberated having two walls. Microspore. Gradually it grows it is the pollen grain. These are two cell stage. Two cell stage. Now, nuclear division. To form eight nuclei. Then, if you go to the structure of out of these eight nuclei, you know, if you go to the ovule now, so you see, this part is called, this end of the ovule is called micropyle. This end is called chalaza. The three cells produced in the chalaza end become
স্যার দেখা যাচ্ছে না।
স্যার দেখা যাচ্ছে না, দেখা যাচ্ছে না।
যাচ্ছে এবার। These three cells are called antipodal cells. Three cells form in the micropyle also. That means these nuclei are bounded by membrane, these nuclei are also bounded by membrane. So here the middle one is called egg, and these two side cells are called synergies, together called egg apparatus. Clear? So in this way we get inside the ovule the megaspore. Who asked me about the archegonium? Do you know, is archegonium required now? Required now?
Rumadash.
What happens? After that, as actually the ovule becomes mature, now this structure, you know what it is called? You can call it, here there are two nuclei only. These two are called polar nuclei. Is it okay? After that, then pollination occurs. একটু মুছে দিতে হবে দেখি Just in the meantime, you see, so this is the pollen tube. What happens here? At 2 cell stage in 60% of the angiospermic plants, pollination occurs. That is, pollen grains are shifted to the anther to stigma. This is called pollination. After reaching their pollen grains by absorbing the stigmatic fluid becomes swollen and its inner wall forming the pollen tube. Is it clear now? That means after reaching the stigma, what happens? I'm showing you. After reaching the stigma, so it's a pollen grain having 2 cells. Like this. Now, after reaching the stigma, it absorbs stigmatic fluid and becomes swollen. And its inner wall is coming out in the form of pollen tube. In the meantime, the generative cell divides to form 2 male gametes. And the vegetative cell becomes degenerating tube nucleus. So, and this structure is called pollen tube. So this is the pollen tube. So in this way we get, that means generative cell divides to form 2 male gametes. Suppose this is the generative cell and we can later. Generative cell divides to form 2 male gametes. So, the 2 male gametes are formed. Here you will get the 2 male gametes. In the center, just before fertilization, only egg is present. Synergids and antipodal cell degenerate The two polar nuclei may fuse to form diploid secondary nucleus. Synergids, before the entry of pollen tube, the role of synergids is to guide the pollen tube to enter the embryo sac. This structure is called, why this is called embryo sac. This is also called embryo sac. Whenever the two male gametes enter the embryo sac, then what will happen? One male gamete fertilizes the egg. This is the egg. To form the zygote. So one male gamete. আসছে কি না আসছে না এই জায়গাটা একটু মুছে দিই আমি পরে আমি এটা একে দেবো একটু মুছে দিচ্ছি one male gamete fertilizes the egg to form zygote. Other male gamete so then after that ovule gets converted to seed and ovary gets converted to fruit. Ovary gets converted to fruit. Eventually inside the fruit there is two embryo. After embryo you will get then a cotyledon is developed, then the new plant. So this is what new plant means. Again this one like this. And from here you will get the flower, and from flower this is the stem. Now you tell me, do you have any problem here in the reproduction in flowering plants? First of all, why it is called double fertilization? Secondly, why endosperm is triploid? And thirdly, why endosperm is triploid? And thirdly,
Sir.
you see, bolo,
So the ovule gets converted to
seed. Ovules get converted to seed. And ovary gets converted to fruit.
স্যার আমার একটা কোয়েশ্চেন আছিল স্যার
বলো
আচ্ছা স্যার এদিকে মাইক্রোস্পোরের পরে স্যার প্রোথালিয়াল সেল ফর্ম হয় না
Basically, you know, these cells are not at all called now prothelial cell. Microspore with a single nucleus, if you in class 12 you will get it. The single nucleus is to form this microspore then divides. An unequal division or dissimilar division occurs, producing one vegetative cell and one generative cell. বুঝতে পেরেছি কি বলেছি?
স্যার আনইকুয়াল সেল ডিভিশন হয়
unequal cell division. The vegetative cell is larger, generative cell is smaller.
ঠিক আছে স্যার।
হুম, ক্লিয়ার? ডাউট থাকলে বলো আমাকে এবার।
স্যার ফ্রি নিউক্লিয়াস ডিভিশন মানে?
Free nuclear division means in a cell, number of cells remains same. Karyokinesis is not followed by cytokinesis. Suppose two nuclei are formed in the karyokinesis. If cytokinesis doesn't occur, then it should be a binuclear cell. Suppose it's a two nuclei, look one thing, the single nucleus. After first division, it becomes two nuclear form, but no cytokinesis occur. So cell number remains same. Then again must be nuclear division. Four nuclear form now. Again few nuclear division. You will get now 8 nuclei.
Sir, what is karyokinesis?
Karyokinesis, the division of nucleus is called karyokinesis, and division of cytoplasm is called cytokinesis. During cell division, karyokinesis occurs first, and it is required to be followed by cytokinesis to form two daughter cells from a mother cell in mitosis, or even in meiosis also to form four. But here you see, if in any cell division karyokinesis occurs, but it is not followed by cytokinesis, then what will happen? A binuclear cell will be formed. Cell number remains same. Daughter cell or the actual mother cell remains same as, but the number of nuclei increases. And such a type of division is called free nuclear division, where nuclei is divided but division of cytoplasm doesn't occur. বুঝতে পেরেছো?
স্যার ভেজিটেটিভ সেল কি?
হ্যাঁ,
স্যার ভেজিটেটিভ সেলটা কি হয়েছে?
the microspores having a single nucleus, when it divides unequally, microspore undergoes unequal division to form two unequal cells. The larger cell is called vegetative cell, and the smaller cell is called the generative cell. So the nucleus is called vegetative nucleus, and this nucleus is called generative nucleus. And in time, generative nucleus will divide to form two male gametes.
Sir,
Ah.
how is triploid primary endosperm nucleus formed?
You know that in the two polar nuclei are present in the middle of the embryo sac, they eventually fuse to form secondary diploid nucleus. One male gamete fertilizes the egg to form the zygote. What about the other male gamete? Other male gamete fertilizes the or gets fused with the secondary diploid nucleus. So one male mate is N, secondary diploid nucleus is 2N. So N plus 2N equal to N plus 2N equal to 3N. Triplets, primary endosperm nucleus is formed. Eventually, from this, gradually, primary endosperm cell and then endosperm is formed. And endosperm is required to provide nourishment to the developing embryo. That's why after being zygote, after being formed, it is found to be in dormant state. Why? So that in the meantime, from primary endosperm nucleus, embryo can be formed. Till endosperm is not formed, then embryonic development cannot be initiated. That's why zygote is found to be a dormant state just after fertilization. বুঝতে পেরেছো এবার?
Yes, sir.
বলো।
স্যার লিখিত যে পোলিন গ্রেনের পরে পলিনেশন অকার, তারপরে স্টেপটা কি?
The pollination occurs means these have to be shifted to the stigma. From anther, pollen grains are liberated and shifted to the stigma. In the meantime, female gametophytic development is completed. After reaching stigma, then what will happen? This pollen grain absorbs stigmatic fluid. Okay.
পলিনেশন অকার্স এই জায়গাটা।
Pollination occurs After pollination, what happens? The stigma, pollen grains are shifted to the stigma, and after reaching there, they absorb stigmatic fluid to become swollen. And then the inner wall of the pollen grain comes out in the form of pollen tube. In the meantime, the vegetative cell becomes degenerate and become present as degenerative tube nucleus, and generative cell divides to form two male gametes. All these things are happening in the stigma. After that, pollen tube reaches the embryo sac carrying two male gametes. One male gamete fertilizes the egg to form the zygote, and other male gamete gets fused with the what? The diploid secondary nucleus to form the triploid primary endosperm nucleus. Eventually, it will become primary endosperm cell, and then from it, endosperm is formed. This is what happens in case of flowering plants. देखो कौन डाउट আছে किधर?
স্যার
বলো।
স্যার পোলিন টিউবের ডিজেনারেটিং টিউব নিউক্লিয়াসটা এটা কি হয়?
নষ্ট হয়ে যাবে। ডিজেনারেটিং শব্দ অর্থ কি? নষ্ট হয়ে যাবে। I will be giving you the questions within 2-3 days, and then within 5 to 6 days there will be an exam. Get ready for this. So after that, chapter 4. Is the animal kingdom. Animal kingdom basically it is from Porifera to phylum Porifera to phylum Chordata. Porifera to Chordata. Now there are different bases taken for classification of the animals. For example, I am showing you. First one, the cellular organization. Then suppose symmetry. Number of germ layers. Nature of xylem. Maybe segmentation. A lot of criteria may be chosen for classification of the animal kingdom. So some of them I am going to discuss here. First of all, what I say is the arrangement of cells. That is the cellular, you can call it arrangement of cell. So you see, first of all, I am to start with this. Based on it, animals are all based on arrangement of cells. Where interpretation is found. 1. এটা সেলুলার লেভেল অফ তার বুঝতে সুবিধা হবে লেভেল অফ অর্গানাইজেশন cellular level organization where cells are formed but tissues are not formed. The animals are multicellular but cells are not differentiated to form tissues. Here Here cells are not differentiated to form tissues. Second, tissue level organization. For example, porifera. In members of porifera, such type is found. Point 1. Man tissue level organization. Here cells are differentiated to form tissues, but tissues are not differentiated further. Okay. Next comes to the found in members of Nidaria and Ctenophora. Next,
স্যার নিচের জায়গাটা দেখা যাচ্ছে না।
হ্যাঁ
স্যার দেখা যাচ্ছে না নিচে দিয়ে।
Oh, tissues are differentiated to form organs. Tissues are differentiated to form organs and but to form organ system. Example found in the members of Platyhelminthes. This is the organ level organization. Last one, organ system level organization. Organs to form organ system found in the members of nemathelminthes to chordata. So from nemathelminthes onwards, we are getting organ system level organization. So it is clear now. You will get tissue level organization, cellular level organization is phylum Porifera. You get tissue level organization in Cnidaria and Ctenophora, that is Hydra, jellyfish, etc. You will get organ level organization in Platyhelminthes like tapeworm. You will get organ system level organization in what is called phylum Cnidaria to phylum what? Chordata. So this is what about the beginning of the animal kingdom, where only level of organization is shown, where cellular arrangement is shown, and based on the arrangement of cells, we get actually 3 types of organization, 4 types of organization. Cellular level organization, where cells are formed but tissues are not formed. Tissue level organization, where cells differentiate from tissue but tissue will not differentiate further. Then the organ level organization, here organs are formed but further differentiation doesn't occur. And in organ system level, you will get the complete differentiation. Suppose system, digestive system, such systems are not properly found in case of organ level organization. Because in organ level organization, you will get the organs, but not the what is called organ system. Is it clear now? In cellular level, you will get the cells, not the tissues. In tissue level, you will get the cells and tissues, but not the organs. In organ level, you will get the organs, but not the organ system. But in organ system level, you will get the organ system. And from here, higher characteristics are found to be developed. Clear? Any doubt? Symmetry and other things. Okay. And in the meantime, you have to prepare yourself for the what is called the test. Syllabus should be biological classification plan.